thanks everyone for joining us for the January 2025 seminar on physical genomics. My name is Benjamin Keene here at CPGE at Northwestern University. As you all know, we're really excited to welcome our speaker today, Dr. Joe Dekker. Dr. Dekker is an investigator of the Howard Hughes Medical Institute and professor in the Department of Systems Biology at University of Massachusetts Chan Medical School. I'm sure most of you are aware his laboratory studies how genomes are folded in 3 dimensions. His work has led to insights into the formation of chromatin loops involved in long-range gene regulation, the organization of the interphase nucleus, the structure of metaphase chromosomes, and general folding principles of genomes. His group pioneered the use of genome folding data for genome assembly, and his lab explores chromosome folding mechanisms in organisms with unusual genome organizations, such as dinoflagellates. Dr. Dekker, thank you for taking the time to share your work with us today. Quick note for our audience, we do Q&A at the end of the talk, so you can either save your questions till the end, or if you want, you can type them directly into the Zoom chat, and we'll address them at the end of the talk. And if you'd like to learn any more about the research going on here at CPGE at Northwestern, you can visit us at physicalgenomics.northwestern.edu. or check us out online. We've got a video archive on YouTube, and we're on Twitter, X, and Instagram. So that's my spiel. At this point, Dr. Dekker, I can turn the floor over to you. Thank you so much again for doing this for us today.
Well, thank you so much for that introduction, and I'm really excited to be part of this seminar series and to present some of the work ongoing in my lab. As mentioned in the introduction, we study a whole range of different things. Today, I want to focus specifically on our work on mechanisms of chromosome folding in mammalian cells, mostly also in chickens, not so much on some of our work on other organisms, but I'd love to invite you to read up on some of that work we do there as well. So today what I wanted to do is to give a brief overview of what we currently think are the main mechanisms of chromosome folding, and then present to you 2 stories that are unpublished, yet they are on bioRxiv, so if you want to study them further, specifically on mechanisms of chromosome folding using the cell cycle as a model system, particularly as cells go in and out of mitosis, which has proven to be a really rich area of study to understand basic mechanisms of chromosome folding in general. So, why is it not working? There we go. So, the main question that drives research in my lab is, how do cells fold their genome? So here on the left, we have a single human nucleus from Irina Solovey's lab, a beautiful image of the human genome. It's folded inside that cell. There are 46 chromosomes. And the question is, how are they folded? What is the 3-dimensional structure of chromosomes? Once we know that, we want to know which molecular machines and possibly other mechanisms, biophysical mechanisms, could be used by the cell to fold chromosomes in that particular shape. And obviously, once we know how that works, we want to understand how folding relates to genome function. Now, when people talk about genome function, one thing that is often immediately thought of is gene expression. But in my mind, there's many functions that genomes have, including DNA replication and particularly chromosome inheritance, how you can inherit chromosomes from one cell to the next. And chromosome folding actually plays a major role in that aspect. And I actually believe much of the mechanisms of chromosome folding are related to that duplication, compaction, and segregation process of genomes rather than gene expression. Now, to study chromosome folding, it won't be a surprise to this audience, we use methods like chromosome conformation capture, or 3C, or variants thereof, to study the folding of chromosomes at increasing resolution. And I'm sure in this series we've heard lots of examples of this, but just to show you a brief overview, what we use in general are 3C-based methods like Hi-C. I think in this talk it's all Hi-C that I will present. And the idea is basically we're mapping interactions between loci, and through a series of molecular steps, physical interactions are converted into DNA ligation products that can be then detected by deep sequencing. And with current sequencing levels, we're going to get chromosome interaction maps like shown here on the right, at resolutions that allow us to study chromosome folding at KB resolution. There are other methods out there that have pushed this even further. We can now map things at nucleosomal resolution for organisms with nucleosomes. And using things like capture-based approaches, we get incredible deep insights into chromosome folding. Today, I will mostly speak about folding at the whole chromosome level, and resolutions of a few kilobases is about as good as we need for that. So what we've learned from these kinds of contact maps, just as a basic introduction, excuse me, is that chromosomes here on the left, we have an interaction map of a few chromosomes with other chromosomes. What we typically see is that intrachromosomal interactions are very frequent. Interchromosomal interactions are less frequent, consistent with the notion that chromosomes form their own spatial territories that are interacting with each other at specific places, mostly active gene regions, but chromosomes tend to stay by themselves. In detail, when you look inside a chromosome, you get this beautiful checkerboard pattern. One of the first features we actually described in Hi-C back in 2009. And this checkerboard represents two types of chromatin that self-associate through homotypic interactions. So when I come back to decomposition, we can identify the positions of these checkers, and red and blue checkers represent active and inactive chromatin domain that in three-dimensional space self-segregate from each other. This is a biophysical process of compartmentalization, and we'll get back to that later. And you zoom in even more, we start to see looping interactions or very precise point-to-point interactions between specific loci. We can see domains of enriched interactions. We can see boundaries and all kinds of complicated structures. Excuse me, I'm still recovering from a cold. So we discover, and others have as well, identified a whole bunch of different structures that can be detected in interphase chromosomes. From such maps and many studies from many people in the field, many may have been on this call, it turns out to form all these complicated structures seen at different levels, both by Hi-C but also by microscopy, you can boil it down to basically having two types of phenomena. First, compartmentalization. This is this checkerboard pattern that we see at the whole chromosome level, also between chromosomes, which is defined as the spatial segregation of different types of chromatin. For instance, heterochromatin and euchromatin, and they are spatially segregated from each other. There might be more flavors of heterochromatin and flavors of euchromatin, but overall, there are two major classes. Now, the process, the mechanism, the basis of this spatial segregation is probably biophysics rather than, say, molecular, in the sense that it's driven by affinities between loci. So euchromatic loci have an affinity for other euchromatic loci, but they try to not be attracted to heterochromatin. And this leads automatically to a separation of phases. On the other hand, there is a molecular machine that makes loops. And not just the loops, all these complicated features I just described, like domains, boundaries, looping interactions between, between specific sites, they are driven, as we now think, and shown mostly by single molecule experiments, modeling, as well as genomic experiments. These are chromosome looping phenomena formed by cohesin and condensin, maybe SMC5-6 complexes. These are molecular machines that actively fold chromosomes. And they form loops and a variety of other structures, as I will highlight in a second. The key point is that there are two layers of driving forces, so to speak, that fold chromosomes. One is a biophysical process of phase separation, and one is a molecular process of active folding machines that bind DNA and fold it. When you think about these two mechanisms, they are very distinct, and this is very important or in this slide for the second half of the presentation. The first is that compartmentalization, driven by phase separation, as I said, is affinity driven. Like B compartments or heterochromatin likes to attract or be associated with other B loci, but not with A loci. So this makes it a very general property, but it's locus independent, meaning it's indiscriminate. Any B domain tends to be attracted to any other B domain. Any A domain is attracted to any other A domain. So it's indiscriminate. All possible BB or AA interactions can happen. There's a quantitative trend for nearby versus far away along the linear genome, but in theory, it's indiscriminate. Can happen both in cis within a chromosome and in trans between chromosomes. This is a very different property when you compare it to formation of loops by things like cohesin and condensin. Those machines make loops by actively extruding chromatin. This is a motor-driven process. So it's an active process. It's not indiscriminate. It happens with loci within certain distance of each other in the linear genome, hundreds of kilobase or so for cohesin, and it's strictly in cis. Loop extrusion doesn't bring loci together in trans. So this is a very important distinction. If you have a phenomenon you see in the genome, these are helpful parameters to think about whether something is in a compartmentalization phenomenon or more an active folding process like loop formation. Okay, so loop formation has gained a tremendous amount of attention in the last several years. Originally, the idea was proposed by many different people over the last decades for various reasons. But the idea is here formalized by Leonid Murney's lab. You have a machine like cohesin or condensin, can bind DNA, and after binding in some way, it will extrude in an ATP-dependent manner a chromosome loop. This was originally a theoretical proposal, but it was subsequently demonstrated in 2018 by single molecule experiments. Here is me doing a single molecule experiment to illustrate the basic idea of loop extrusion, but actually Kees Dekker and his group were the first to show with condensin, yeast condensin, that it can actually, in single molecules under the microscope, you can see it make loops in an ATP-dependent manner. I think this experiment was really key in steering this field in the new direction of loop formation by these complexes. So it turns out this single process of loop extrusion can explain lots of different things we have seen in high C. So imagine the following scenario. This is from Leonid Murney's lab. You have a chromosome here. It's a polymer. Cohesin or any other extruder, here we just call it cohesin, can bind anywhere along this chromosome, start making loops in cis, but the twist here is that it will stop at certain sites bound by, for instance, CTCF or any other complex on the chromatin that has the potential to block loop extrusion. When you simulate such a system, just by polymer simulations, you get a structure of chromatin that doesn't look too informative, but if you then predict what high C would look like when this system works in living cells, you can see the predicted high C map looks very much like the high resolution high C maps and micro-C maps that we and others have generated for real cells. So what it basically means is that the singular process of loop extrusion can really explain loops between sites where cohesin gets blocked, like CTCF sites, line-like features, often anchored at CTCF sites, topologically associated domain-like structures with enriched interactions between CTCF sites, and so on. So a single process can actually explain lots of different features in high C that a priori we didn't know whether a single process or maybe multiple different processes would drive that. Now, this model has, of course, been updated quite a bit over the years. Here, I just show you an example of a proposal we made a few years ago, but Wouter de Laat's lab had a very similar independent study showing similar things, where this idea that cohesin can just load anywhere is probably not entirely correct, although it does load probably everywhere. But it has certain preferred sites for entry onto the chromatin. This could be enhancers. And there's some work from Leonid Murney's lab and others that suggest something similar in early development, that the first places where loop extrusion starts is at regulatory elements such as enhancers. Cohesin can then, or any other extruder, can start moving along the chromosome, maybe stop or briefly pause at transcription start sites. Then it gets into the gene where I think it probably is quickly pushed out by RNA polymerase. Ed Bannigan has shown some nice work with Jan Michael Peters and Leonid to show that RNA polymerase and cohesin are not very compatible with each other. When cohesin then reaches the 3' end of active genes, it gets correctly unloaded. When it occasionally does go further, it will ultimately get stopped by CTCF sites. But what this slide what I want to illustrate with this slide is that cohesin is actually highly dynamic along the chromosome. It's moving, it is loading and unloading and pausing and stopping and being blocked at various places. And the end result or average result of this, what we call cohesin traffic pattern, is what we finally see in a high-C map. But what that map underestimates in a way is the remarkable dynamics of the process. And second, that the entire process is related to cis elements that we typically associate with transcriptional regulation, like enhancers, start sites, end sites of genes, insulators, and so on. So this already illustrates this close connection between these folding machines, loop extrusion, and gene regulation, although many questions remain. I'm not going to spend a lot more time on it today. I just wanted to highlight this because this is an example where there's this close connection between chromosome folding, cis elements, and gene regulation. Okay, now it turns out that actually when cells cycle through the cell cycle, their chromosomes change dramatically. So what I talked to you about up to now is mostly interface. The interface nucleus is rather diffuse in the microscope. We see this beautiful checkerboard pattern of compartments in interface. But by metaphase, we showed this a number of years ago, all these structures disappear. In the microscope now, the chromosome looks highly structured. It's a beautiful X-shaped structure, but in high-C, this map looks remarkably devoid of features, which is still, I think, a remarkable thing when you think about it. So as these chromosomes change their shape during the cell cycle, again, we could imagine that we would have dramatically different mechanisms folding these chromosomes. But again, it turns out you don't need new mechanisms. You just need a few mechanisms to make all these different conformations. So both for interface and metaphase, similar mechanisms are used for folding these chromosomes. And that's what the first part of my talk will be. And that is that the first story that I will talk about is I call it rules of engagement for chromosome folding machines. And again, the theme there is that the mechanism of loop extrusion and loop formation is applying to all chromosomes in interphase and in mitosis, but they are being employed in different ways to fold either cell chromosomes into interphase or into mitotic chromosome. The second part is a shorter story where I will talk about how information for chromosome folding, that is cell type specific, can be inherited through mitosis when chromosomes are otherwise folded in a cell type invariant manner. Okay, so let's take a look at mitotic chromosomes now. Mitotic chromosomes, as I said, are the classical X-shaped structures, well studied for decades. And here we have a beautiful picture from Uli Lemley's lab and Keisu Mishima, where in blue is the DNA, and in purple we have condensin, which folds, makes loops during mitosis. So a question has been driving research in my lab for quite a while, actually from ever when I started my lab, is how is the DNA folded inside these mitotic chromosomes? And it turns out the mitotic chromosome is almost exclusively made by loop extrusion. It's the one mechanism that drives chromosome folding. So that is actually, if you're interested in loop extrusion in general, studying how cells can make mitotic chromosomes is really a fantastic model system. Now, what is interesting about mitosis is that to make a mitotic chromosome, there are multiple different loop extruding machines acting on the chromosome simultaneously. And that raises important questions as to how these loop extruding machines can work together or not, or at least deal with each other as they fold the mitotic chromosome. So as I will point out in a second, is that there are, as I said, there are multiple different loop extruding machines acting. And that is, we have still cohesin that normally is active during, say, G2, just before cells enter mitosis. But then we have two condensins, condensin 1 and 2, but we also have cohesin that holds the sister chromatids together. So there are at least 4 of these SMC complexes, bound loop extruding complexes, working together. So what we know about the mitotic chromosome for quite a while, from Uli Lendi's work originally, that was later confirmed by Heisey by us, is that the basic architecture of a mitotic chromosome is an array of loops. It's a very stacked and compressed array of loops, and it's made by the condensin 1 and 2 complexes. These loops in human or mammalian cells are typically around 80 kb in size, but a key difference for cohesin is it doesn't get stuck at any particular places, so the loops made by condensin are not positioned at any specific DNA sequences. So that's important for Heisey, because in Heisey, we won't see any recurring positioned loops at certain sequences. And what the Heisey map shows is just the overall pattern of a randomly positioned loop array. A few years after we showed that work that I just highlighted, we showed that actually the mitotic chromosome is a little bit more complicated. And that is, first, the loops themselves are of 2 types. We have large condensin 2 loops, here in red, made by the condensin 2 machine. These loops can be hundreds of kilobases, 400 or 500 kilobases, depending on the organism. But each of these big condensin 2 loops is then split into smaller loops by condensin 1, which basically makes a set of nested loops. And these condensin 1 loops tend to be around 80 kb, and that's the loop size you typically see in an imaging experiment. Second, we show that the loops are not randomly protruding from a core of condensins, excuse me, but rather the loops are rotating somewhat, not entirely, somewhat in a genomic order around, in a helical path around the central core of condensin. So the whole chromosome has some intrinsic helical organization. Okay, so the main question that we're going to focus on in the next couple of minutes is what happens, how can cells convert this interface structure that I spoke about quite at length with compartments and cohesin loops, how can that structure be quickly, and I'm talking about 10 minutes, how can the cell converge that interface structure into a metaphase structure where the chromosomes now form a condensin-driven array of loops with this helical aspect? And again, I still find it amazing how efficiently cells can do this. And as I said, during this transition, during prophase, there are 4 of these complexes working on the chromosome simultaneously. They all have SMC coiled coil proteins and then various subunits. They differ in that regard. So we have cohesive cohesin, as I call it. That's, of course, the cohesin complex that led to the name cohesin, is this ring-shaped complex that holds sister chromatids together. We don't think it's actively extruding, but it may have done in the past. But by the time we're in prophase, this is a complex that holds pairs of sister chromatids together. We still have extrusive cohesin, meaning the cohesin complex that's actively making loops in cis, like I drew before. That's still making loops in early prophase. And then as cells enter prophase, condensin 2 becomes active. And this condensin doesn't act in interphase, but starts making these bigger condensin 2 loops that drive mitotic chromosome formation. And then by later prophase, early prometaphase, condensin 1 comes in and starts making these nested loops, loops within loops. So these 4 very similar complexes start acting on the chromosome. Now, I won't show you because I won't have time. We were able to measure the in vivo speed of loop extrusion for condensins. And we think other people, Alistair Buttinger and others, Anders Hansen, have measured and estimated loop extrusion speeds for cohesins. But in all these experiments, these are in vivo speeds. The speeds are between, say, 2 and 3 kilobase per second, which is a remarkably high speed. Because, like, if you think about RNA polymerase, that has a speed of about 1 kilobase per minute. These extruding complexes make loops 2 to 3 kilobase per second. And this is very similar, actually, to what Kees Dekker and others have measured in vitro. So I have this mental picture in my head. We have a linear chromosome, which is like a single-lane highway. And on this highway, we have these SMC complexes racing with a high speed, 3 kilobases per second, but they race in both directions on this single lane highway. So they are going to run into each other. And then what will happen? So the main question that we had is we have so much work on these chromosomes. These complexes are running around. They're going to collide. And how are such collisions being resolved? And I will present some work. There's a lot more in our preprint, but just some aspects of that work to illustrate how such encounters between SMC complexes are resolved. And they're resolved following a set what we call rules of engagement that determine how specific pairwise encounters are being resolved. So let's go back to the model of a mitotic chromosome. Our model in 2018 was that the chromosome is a nested loop of arrays that has a helical aspect. But the way we calculated these models, we just focus on one sister chromatid. But in reality, of course, there's a second sister chromatid that has a related architecture. And these two sister chromatids are held together by cohesive cohesins that we know are localized somewhere in between or at the interface between the two interacting sisters. By this time, late prometaphase, any cohesin that was making loops during interphase has somehow been removed. So the question really is, how does this arrangement of condensin, nested loops, cohesive cohesin in between the sisters, and loss of extrusive cohesin, how is this accomplished within 10 minutes? To study such a fast-moving process using a bulk assay like Hi-C, we need super high synchrony of cells. So for this, we used chicken DT40 cells in collaboration with Bill Earnshaw's lab. These cells were engineered to express a CDK1 analog sensitive allele. This CDK1 is the key kinase driving cells into mitosis. This allele can be inhibited by a nucleotide analog, so that when you treat cells with this analog, all cells will be blocked in G2. And you can just simply wash out the analog, and then cells will, as a whole bulk assay in lockstep, move through prophase, prometaphase, and onwards, and with a high synchrony. So we can actually do minute-by-minute experiments by Hi-C to study chromosome folding as interphase structures disappear, mitotic chromosomes appear. We combine this with a series of cell lines made by Kumiko Samejima in Bill Earnshaw's lab, where we can deplete using inducible Dekkerons 1, 2, or 3 of the 4 SMC complexes present during prophase. So for instance, we can just remove condensin 1, or just remove condensin 2, or both, or we remove only cohesin, or we remove cohesin and condensin 1, and so on. All the different combinations to test what is the defect when you lose one of these complexes, but also what can the cell do if only one of these complexes is present? And I'll just show you an example of the kinds of experiments we did. So here we have some images of cells on top with control cells arrested in G2. We have a beautiful interphase nucleus. We release these cells from the CDK1 arrest, and the cell starts entering prophase and prometaphase. This is about 30 minutes in at the last time point, but here about 10 minutes you start to see nice chromosomes. This just shows how fast mitotic chromosome formation is. The bottom row shows cells in which we depleted SMC2. SMC2 is a part of both condensin 1 and 2, so these cells have no condensins. These cells actually do enter mitosis. The chromosome does compact, about twofold compaction in volume, but they never form chromosomes. This is not unexpected. We know condensins are essential for making rod-shaped chromosomes. Now, on top is just an example of, you see the microscope. Here in the bottom is what we see by Hi-C. Hi-C maps tend to be symmetrical, so I just show 2 heat maps of Hi-C basically in one square. The bottom is the wild type. In G2, we see beautiful checkerboard in wild type cells. The top is in G2 cells where we deplete condensin. Nothing changes. We don't expect to see a change. Condensin is not required or involved in making G2 chromosome structures, but you see beautiful compartmentalization. As cells enter prophase, we can see 2.5, 5. By 10 minutes, this checkerboard pattern that defines compartments is completely gone. 15 minutes, and then by 30 minutes, we start seeing here this second diagonal, which which represents this helical aspect of mitotic chromosomes. So this just shows by high C the same thing as we saw on the top. You disassemble the interphase nucleus and you form a mitotic chromosome. On the top, high C part, we see what happens in the absence of condensin. In the absence of condensin, the interphase structure lingers on for a very long time. Ultimately, it seems to fade away, but the cell has a problem to disassemble the interphase nucleus in the absence of condensin. So it already shows clearly you need condensin for disassembling, not just making a mitotic chromosome, but for disassembling the interphase structure. Now, what happens at more local events like cohesin-mediated loops? So here we focus in high C and look at more detail at some contact maps. And here in aggregate at CTCF looping interactions that we see in wild-type cells. And again, in G2 cells, you can deplete condensin here on the top map, no condensin, with auxin. We can see CTCF loops are there, just like in wild type. Condensin doesn't do anything with cohesin loops in G2. But as cells enter mitosis in wild type cells in the bottom, we can see that after 5 and 10 minutes, CTCF interactions between pairs of CTCF sites mediated by cohesin disappear. But in the absence of condensin, that doesn't happen. So in the absence of condensin, the cell has a problem removing cohesin-mediated structures. We can quantify this. Ultimately, these structures, like dots, they do disappear, but much slower in the absence of condensin. So can we model this phenomenon? The idea is that condensin somehow actively speeds up the resolution of these cohesin-mediated structures. So the way we imagine this could happen is in the following 4 different scenarios. Here we start out with a simulated cohesin array of loops. This is like an interface simulation. We have loops between CTCF sites where cohesin stalls. Now, if these cells in the simulation enter mitosis, condensin here in red starts also making loops. And this condensin machine will at some point run into cohesin. And now we have an encounter along the chromosome. 2 different loop extruders, cohesin and condensin. How is this resolved? We've considered, this is really Samir's work in Leonid's lab, we considered 4 different scenarios. First, condensin could actively unload and remove cohesin and then keep extruding. Or it can just push it in front of it and keeps extruding. Those 2 will be very difficult to tell apart. It could just step over the cohesin and make something like a Z-loop that has been shown that condensin can make in vitro. And now we have this more complicated overlapping loop scenario. Or the 2 machines run into each other and then just both of them are stalling and don't know what to do. When you simulate this, the outcomes are quite different. In order to reproduce what we see in experiment, namely the loss of CTCF loops and cohesin-mediated structures, only 2 models will work. It's unloading, condensin will unload cohesin, or it will push it. These 2 are difficult to separate, but we have proteomics data to show that condensin actually facilitates unloading of cohesin complexes from bulk chromatin by proteomics. So we like to think that this is an evidence to suggest that condensin can unload cohesin. Here's another example where we zoom in at some other places in the simulation, and we can see unloading and pushing will remove all these dot-like structures that are normally made by cohesin. So that brings me to my first rule of engagement for SMC complexes. When a condensin during prophase encounters an extrusive cohesin, it will remove that extrusive cohesin and it will keep extruding. Second, when we look at prometaphase cells, we can see here, for instance, here's an example, 2 sister chromatids. In blue and in purple here we have condensins, but there is cohesin still on the chromosome and is located in between the 2 sister chromatids. If you take lots of cross-sections through pairs of sister chromatids, you will see the DAPI, the 2 sisters squished together. In the core of each sister, we have condensins at the base of the loops. We expect it to be in the core of the center core of the chromosome. But cohesin is in between, and this is cohesive cohesin. these cohesin complexes that hold the pairs of sisters together. And the question we had is, how did it end up this way? How does cohesive cohesin end up in between the masses of the sister chromatids? And again, we use polymer simulations. So again, it is some years' work. We start in G2 when we have two sister chromatids now held together in this green complex here, a cohesive cohesin. And cells enter mitosis, condensin starts making loops in cis, only along each sister chromatid. And at some point, such a condensin is going to run into this green cohesin complex. And the question is, what will happen? Here we consider two scenarios. One is nothing happens. The condensins stay stuck at this cohesin, and now you end up with an array of loops made by condensin, but the sister chromatids are connected at the base of the loop. Alternatively, condensin can step over such cohesive cohesin. Now we have loops along each sister chromatid, but the sisters are now connected to each other through the tips of the loop. We can do this in real simulations. These are, again, simulations from Samir. And the key difference between these two scenarios is where cohesin is with respect to condensin. If condensin gets stuck on cohesin, the two sister chromatids are connected at the bases of their loops, and condensin and cohesin in an average cross-section will tend to co-localize. Whereas if condensin can step over cohesive cohesin, we end up with two axes of sister chromatids. The bases of the loops are separate from each other, and cohesive cohesin is located in between these axes in the tips of the loops. This is what we see in experiments. So at least by prometaphase, we know the sister axes have split with cohesin in between. That can occur when condensin somehow is able to bypass cohesive cohesin. And that brings us to our second rule of engagement. The first one was when condensin runs into an extrusive cohesin, it will remove it. But when it runs into this other type of cohesin, a cohesive cohesin, condensin can step over that cohesive cohesin. And now cohesive cohesin ends up inside the loop. This model does not rule out that transiently during this encounter, condensin and cohesin do co-localize, which leads at least transiently to joining of the sister axes, which is exactly what Nancy Kleckner has shown in her work during early prophase or later prophase stages as well. So that would propose that at least there is an initial pause and then a bypassing. Finally, we did a lot of work studying mitotic chromosomes made by only one or only two condensing complexes in the absence of cohesin. And I won't show you all the things we've learned from this. It's in the preprint, and you are encouraged to read it. What I want to highlight here is just some remarkable high-C data we were able to obtain if we made chromosomes with only condensin 1 and 2, condensin 2 only, condensin 1 only, or no extruders whatsoever. I just want to highlight one particular contact map. This one here, this is a mitotic chromosome map made by cells only expressing condensin 2. And look at this remarkable map. It looks almost like synthetic data, but I promise you, this is real data. We have a primary diagonal we always see in high C, a secondary diagonal, third diagonal, and for some long chromosomes, we can even see a fourth diagonal. We now understand where all these multiple diagonals come from. It has to do with the helical coiling of the chromosomes. Anton Goloborodko in Vienna did polymer simulations to make models of chromosomes that could reproduce these high-C data. And just briefly, what he does is he takes a chromosome, treats it like a long polymer that fits inside the shape of a mitotic chromosome, and then imposes various constraints on it, like is there a helical constraint, are there loops of certain sizes, and so on. And he can reproduce, these models can reproduce the high-C data here, for instance, for a condensin 2 only chromosome very, very nicely. And again, we end up with beautiful new models that update our model we published in 2018. We derive important conclusions from this, like the helicity we originally drew as quite regular. It doesn't have to be that regular to look regular on average, but in single cells, it's not very regular. We think there's some spacing between the loops, and I think Fitz-Hubert's lab has shown that as well in barley. We learned things about how cohesion between the sister chromatids can help constrain this helical path and so on. But the key thing I want to emphasize that we learned from these models is that in all these models, the Condensin loops that we model have to be consecutive in order to reproduce both the Hi-C and the imaging data. What does it mean for loops to be consecutive? It means that one condensin stops, one loop stops where the next one begins. They don't overlap. And that can only happen when condensins stop extruding when they run into each other and do not pass or unload one another. So that is an indirect inference from the model. So probably maybe the weakest rule of engagement, but that's our third rule of engagement. The first one is when condensin encounters an extrusive cohesin, it will remove it. When it encounters a cohesive cohesin, it can ultimately step over it, leading to the separation of sisters in prometaphase. But then when condensin runs into another condensin, they just stop and stay. So these are three rules that define encounters between the different loop extruders during prophase. They're different for different types of encounters. But what I found remarkable about this is that when you combine these rules with the local action of loop extrusion, which is kind of like agnostic as to where the condensin is or cohesin is on the chromosome, it acts on a very local scale. But when you impose these rules about what happens when they run into each other, this complicated arrangement of nested loops by condensins, cohesive cohesin in between the sisters, and loss of extrusive cohesin, it automatically emerges by self-assembly. So it's I think, a very elegant way of thinking about how local acting machines like loop extruders can work together to generate a structure at a micron scale, a length scale is much bigger than the local molecular action of these machines. No doubt there's going to be refinements of these rules and exceptions, and also new rules. And these machines can run into all kinds of other complexes, and they may have different outcomes. For instance, Romain Kozul's lab in Paris has shown very nicely that when extrusive cohesin runs into a cohesive cohesin, so the two different cohesin complexes, when they run into each other, they stop. So that's a very different outcome than what we have seen when condensin runs into a cohesive cohesin. So there are all these different outcomes that can be studied. And the question, of course, is molecularly, how does it all work? We don't know. This is the longest part of the talk, I promise. It was run by this group that we call the Chicken Group. It's really a fantastic collaboration for almost a decade now with Bill Earnshaw's lab, Leonid's lab, Anton's lab, and lots of key people, Kumiko and Johan, who are really the drivers, and Fernanda, between the different labs, and Samir, to put this all together. So it's been a fantastic collaboration. Now, I want to move on with a shorter talk at the end, where we go and say, okay, we have now made a mitotic chromosome, but when the cells divide, that mitotic chromosome has to now be disassembled, and the cell has to rebuild an interphase structure. Now, this is not just the inverse of what happens when you go into mitosis, for various reasons. For instance, when cells are in interphase, the conformation of the chromosome has certain rules, so to speak, but it's distinct and cell type specific. Which loci are in the A compartment or engaged in the CTCF loop is different between different cell types. So both G2 but also G1 structures are cell type specific. The mitotic structure, at least the structure seen by Hi-C in imaging, seems to be rather invariant between cell types. It always forms an array of loops. The loops are not positioned at any specific sequences. So the question is, how does the cell know how to disassemble this structure and rebuild an interphase structure that needs to be the same as the one they had in the previous cell cycle to maintain cell state? Lots of things have to happen as cells exit mitosis, and a few years ago, we and Gerd Blobel and a few others have started to study this process of rebuilding the G1 interphase nucleus. So as I said, in G1 or interphase, we have beautiful checkerboards that represent the compartments. We have loops and tats and so on. Then you go into mitosis. I just described that process. You form this loop array, a nested helical loop array. But then as cells exit mitosis, this is data from a paper we had a few years ago, that structure has to be rebuilt, the interphase structure. And actually, it takes remarkably long compared to the making of a mitotic chromosome, which can be done in 10 minutes. This occurs within about several hours, particularly compartmentalization, this checkerboard patterning, takes quite a while, a lot of time to get back. Whereas loop extrusion features come back earlier, and they are established pretty quickly. So the question that Alana Schooley in the lab had is what kind of instructions do you need for this? Like what proteins and so on do you need for telling the cell how to fold an interphase nucleus? And where are they during mitosis? How are they inherited through mitosis? So she imagined you could have factors like histomodifications that could be determined. They are on the chromosome. So the idea would be there are probably factors and proteins and information carried on the chromatin during mitosis. It's not being used to instruct the cell how to fold it. It's just universally folded by condensins. But the instructions are still there on the chromosome. But you could also have factors that actually are inherited through the cytoplasm. They dissociate from chromosomes, and we know many transcription factors, CTCF, RNA polymerase, chromatin remodelers, they all dissociate from mitotic chromosome, now are in the cytoplasm. So in the next G1, they have to be coming back from the cytoplasm, back on the chromosome. And what she wanted to know is, can I tell them apart? Can I know which kind of factors that are needed for chromosome folding are on the mitotic, inherited through the chromatin, or are inherited through the cytoplasm? And the idea she had was actually very creative and ended up being very interesting. What if she would block nuclear import during exit from mitosis so that any factors that are inherited through the cytoplasm, they're still there, but they cannot gain access to the chromosomes from telophase or so onward when the chromosome is still highly compacted. So basically, it's a mechanical barrier to prevent all these cytoplasmic factors, whom we don't know yet, prevent them from gaining access to the chromosomes. And then we can do high C and ask, okay, what can the cell do? The chromosomes are now inside this impermeable nuclear envelope without nuclear import. What kind of chromosome folding can happen? That must require factors that are inherited on the chromatin. And which things cannot happen? Those would be things requiring factors from the cytoplasm. Okay, so the way she did this was she used cells made by Fasa Lisa in Mari Dassov's lab at NIH, really fantastic collaborators who shared these cell lines early on with us and helped us do these experiments. So the idea is very simple. We take prometaphase cells, they're normal, everything is fine, they have mitotic chromosomes, and then we induce these cells to degrade RAN-GAP or NUP93. These are two essential proteins involved in nuclear import. One is RAN-GAP is important for the gradient of the GTP-GTP gradient across nuclear pores required for import and export, and NUP93 is an integral component of the nuclear pore. So in the absence of these proteins, when the proteins are removed, Alana will release the cells into anaphase. Cells will divide, and the nucleus will quickly reform around the telophase chromosomes, but now without nuclear import. These chromosomes will stay physically isolated from the cytoplasm. She can show here by imaging that in the depleted cases, we can deplete all the RAN-GAP or the NUP93. By Western, we can see these depletions are really, really good. These are really efficient diagonal lines. The cells actually exit mitosis just fine without these proteins. Here we have a control cell, here in prometaphase, anaphase. By telophase, they no longer have things like phosphorylated histones, everything is fine, and then you form beautiful G1 nuclei. For instance, cells in which we deplete RAN-GAP during prometaphase and then release cells, the timing of events is just fine. The cells can go through anaphase, telophase, early G1, they form a nuclear envelope, and everything is fine. The only difference is that the nuclei that ultimately form are really tiny compared to control nuclei. But the kinetics of nuclear mitotic exits is fine. As I said, by imaging, we can see some morphological differences, actually. So in control G1 cells that have exited mitosis, we form nice nuclei. There's a strong nucleolus in this particular one. But in the absence of nuclear import, either in the RAN gap depleted cells or NUP93 depleted cells, the nuclei look dense and very small. We quantify it here on the right. The chromatin never really decondenses. But chromosomes are gone. They no longer form mitotic chromosomes. And actually, we can even see in the nuclear envelope that in a RAN gap depleted cells, you do form nuclear pores. There's just no nuclear import. In the absence of NUP93, you don't even form nuclear pores. Vasilisa then did a really important experiment. I won't go through all the details to show that cells actually cannot import anything into these nuclei. So in this case, she depleted RAN gap or NUP93 during prometaphase arrested cells, release the cells, and use an MBP NLS reporter gene, a fluorescent reporter protein fused to a nuclear localization signal, to show that in control cells, after about 30 to 60 minutes, they started importing proteins from the cytoplasm, but in these depleted cells, there's absolutely no nuclear import. So this genome is sitting there completely isolated from the cytoplasm without nuclear import. Okay, so what kind of structures can reform if cells can fold in the absence of nuclear import? So here on the left, we show two chromosomes. This is the G1 after mitotic exit, control cells. They form nuclear territories. If you look inside a chromosome, we have a nice checkerboard. They form compartments. If you zoom in, you see loops made by cohesin. They're mostly short range, but not long range, over tens of megabases. This is a long range contact map. Loop extrusion doesn't reach that far, so we don't see that. However, in the absence of nuclear import, some surprising results were obtained. First, you form nice interphase chromosome territories, a really prominent checkerboard pattern, very strong compartmentalization, just fine without any instructions from the cytoplasm. But when you zoom in more closely, we don't see the local contact map that we see in control cells, which is dominated by these CTCF interactions. What we see instead is a different type of compartmentalization. Small domains start interacting with each other over short distances, but also over very large distances, tens of megabases, and even in trams with other chromosomes. What are these loci? It turns out these loci, we call them microcompartment domains. Those are active chromatin in terms of attack-seek. They're open chromatin. They're marked with K27 acetyl, K4 trimethyl. And it turns out, if you look at cis-regulatory elements, these are little stretches of the genome, 10, 20, 30, 50 kb in size, full of all the regulatory elements and promoters that are active in this cell type. And what is happening is that all these little patches of active chromatin starts to interact with each other indiscriminately in cis and in trans. And again, we can show this more clearly. We show strong enrichment of promoters and proximal and distal enhancer elements are enriched inside these interacting loci, and that's even enriched compared to chromatin, that's active chromatin in general. They are marked by a high level of attack-seek, K4, K27. They are depleted for inactive marks, like K27 trimethyl, and so on. So it's basically all the active chromatin. Now, we think that because they are interacting indiscriminately in cis and in trans, we like to think that these are a compartmentalization affinity-driven process. And the fact that they can happen particularly strongly in nuclei without nuclear import means they must be driven by factors that are already carried on the mitotic chromosome. So that smells like some kind of a bookmarked sites that are already bookmarked in mitosis. So to test this, Alana did attack-seek, identified lots and lots of peaks, and classified them into various types. The most common attack-seek peak seen in the genome are peaks that are seen only in wild-type G1 cells. And these are sites that are open in G1 and closed in mitosis. But they do not reopen in the absence of nuclear import. So that means they are requiring information from the cytoplasm to get reopened. But then there is a large category of sites that are active, that are in open in mitosis, in G1 in wild type, and in G1 in control cells. Now, it turns out if you look at these, if you just look at these microcompartment domains that are starting to interact with each other, they are strongly enriched to the genome-wide compared to genome-wide for those sites that are mitotically bookmarked. So those domains have ataxic peaks that are open in mitosis and in G1, and they don't really need nuclear import to stay open. So that category is strongly overrepresented inside these microcompartment domains. In fact, all microcompartment domains have at least one such peak. So that means that these sites are bookmarked. They remain open in mitosis. The information that tells the cell, this is an open site, I'm a microcompartment domain, that information is present on the mitotic chromosome and inherited through mitosis. Now, what we were somewhat concerned about is that we are discovering this folding structure in a mutant, basically. So it could just be an aberrant structural state that normally never happens in normal cells. So to test this, Alana did a very clever experiment. Actually, it's very hard to do this. Remember in the first part of the talk, I showed you how we set up a system that we can highly synchronously collect samples in prophase and prometaphase and so on. It's very hard to do that during mitotic exit. So she came up with a sorting strategy to specifically sort cells and populations of cells enriched in telophase or in cytokinesis. And it has to do with the shape of the DNA. I won't go into much detail about it. But she could look at very early stages of mitotic exit by Hi-C, with or without nuclear import, and see what she found. Surprisingly, what she found is that in control cells on the top, in prometaphase, we have the typical prometaphase structure, just a single diagonal. What she discovered is that the same maps we see in the mutant cases, the RAND-gap depleted cells, occur very transiently during telophase and cytokinesis. So during that stage, we see the same thing. This compartmentalization appears between those microcompartment domains before we have loop extrusion features made by cohesin. This structure transiently pops up and then disappears and gets replaced by the normal G1 state. Now we see cohesin-mediated loops between CTCF sites, and that original compartmentalization signal is lost. In the case of the RAND-gap depleted cell, the same thing happens. We have beautiful mitotic structures. By telophase and anaphase cytokinesis, we start making this microcompartment domain pattern, this grid of interactions between them. It shows up again, but then it stays and gets stronger and stronger and stronger, which allowed us to initially discover it in G1 in these cases. But this is a normal transient intermediate state that happened during cytokinesis. So from these experiments, we concluded two things. First, there must be two waves of chromosome folding. The first one is driven by chromosome intrinsic factors that leads to this initial clustering of these small active genes and regulatory elements. It's indiscriminate. And then a second program comes in and it modulates this first state, and now we end up with the normal interphase state. And that second folding program doesn't run when you have no nuclear import. So that second folding program that modulates the first one must be coming from the cytoplasm and driven by cytoplasmic factors. Needs nuclear import. Now, we now know what at least part of this second program is, and I'm almost done, I promise. If we look at contact maps control or without nuclear import, we see that in the absence of nuclear import, we have this grid of these indiscriminate interactions between active loci. In control cells, we form TAS and loops and CTCF-mediated structures. So what we think is actually happening is that in control cells, cohesin comes in and starts modulating by loop extrusion this pattern of compartmentalized interactions between active loci in such a way that now the interaction pattern between these active regions is no longer indiscriminate. It now has to obey the laws of loop extrusion so that you lose a lot of these indiscriminate active-active interactions that we see in the RAND-gap depleted cells. They get depleted when cohesin comes in because they would happen across a strong insulation boundary. And indeed, we know that in the absence of RAND-gap, cohesin stays in the cytoplasm. So whereas In control cells, you start forming CTCF loops very quickly from telophase onwards and get very strong in G1. Between CTCF sites, is driven by cohesin. In the absence of nuclear import, there's a little bit happening in early telophase, which might be cohesins coming in before the nuclear envelope is sealed, but you don't form these cohesin structures. So again, as I said, what we think is happening, loop extrusion is putting additional constraint on these otherwise indiscriminate interactions between, say, enhancers and promoters, so that now we keep the ones within extrusion domains or topologically associated domains, but we are losing longer range interactions because they basically get prevented by blocking of loop extrusion by CTCF. So what is more out there, this is just one machine coming from the cytoplasm. So it's probably just one of several processes that starts to become active during mitotic exit, and it needs to be imported from the cytoplasm. As I said earlier, we know that most of the sites that open up again, as we see by ATAC-seq, the majority of them are only seen in G1 cells, not in mitosis. And in order for them to open up, you need nuclear import. So there are factors coming in from the cytoplasm that had to open up these sites. These could be distal enhancers. To start to identify, this is a little open-ended end for this seminar, we started to do proteomics to identify which proteins fail to make it to the nucleus in the absence of nuclear import during mitotic exit. And for this, Alana did a straightforward Silac experiment where she makes nuclei in control cells exiting mitosis or from cells in which nuclear import is depleted. We mix them, we do proteomics to see which proteins are missing. And it's quite remarkable, all the things that don't make it to the nucleus in this experiment. The entire cohesin complex, all the subunits are depleted. Many components of RNA polymerase, we know these nuclei actually don't transcribe anything. Lots of chromatin remodelers, probably that need to open up those ATAC-seq peaks. They are not there. But some other things are still there. The mitotic machine is still there. Should have been depleted, like Condensin 1, still there. Failed to become exported. But also some heterochromatic factors are actually enriched. Polycom, we know polycom proteins get inherited through the mitotic chromosome itself, not through the cytoplasm. So this is just a starting point for Alana's own research going forward. She's on the job market to explore the various machines that have to come in. They also rule out lots of things that we thought were involved in chromosome compartmentalization, like RNA polymerase transcription and so on. All these machines are not even in the nucleus, yet the chromosomes compartmentalize just fine. So compartmentalization is mostly driven by things carried through mitosis on the chromosome. Okay, so to wrap up, interphase chromosomes, we think that it's really folded by two distinct mechanisms. Loop extrusion, transient folded initially driven entirely by chromosome intrinsic affinities. And then a second program comes in, mostly loop extrusion and probably transcription machinery, that then modulates this first folding state to make it into a mature interphase state. So nuclear import deficient cells was kind of like a bizarre cell line initially to begin with, but what it allowed us was to identify two mechanisms, or two folding programs that drive interphase chromosome conformation and to study their interplay, which I think was really key to understand the final interphase structure. So again, this work was done mostly by Alana, collaboration with Vasilisa, and in the later stages during data analysis, Sergey was an absolute key member to contribute to this project. So I just want to end up one point making here. I said we've been studying chromosome folding through the cell cycle for many, many years, and it turned out to be a really rich system to study basic mechanisms of chromosome folding. Because chromosomes change their shape so much, it allowed us to really study very basic concepts of chromosome folding. So finally, I'd like to thank my lab, my funding agencies, and all of you for your attention. Thank you.
Thank you so much. That was fantastic. It's 1:09. If you've got a few minutes for questions, if you'd be willing to take any that, like we've got a couple in the chat already.
Sure. You want me to read the question or?
Sure, if you don't mind, it might be easier if you just read
Yeah,
it.
sure, sure. So Lou Bai asked, thank you for the talk. Does the bookmarked microcompartments bind to specific transcription factors? That's really an excellent question. We don't know the answer to that question. I know many factors dissociate from chromosomes during mitosis, but not all. There are several factors that are surely probably marking bookmarked sites. They could be bookmarked for other reasons. They could be bookmarked because they have histomodifications that don't go away. And as cells exit, those sites are immediately accessible again for factors to bind. But these factors somehow need to either be on the chromatin or need import. We don't know. It's been very tricky, actually, to study transcription factors during mitosis. But I think there is certainly a set of factors that remain bound to sites. It's also somewhat cell type specific. We have a story in preparation where we find that, for instance, CTCF, we've shown in differentiated cell lines, is mostly dissociated from chromosomes, but in other cell lines, particularly pluripotent cells, there's a lot of CTCF still bound to sites. So this requires a lot more work, but I think what I hope is that these kinds of experiments that we're doing can identify factors that really have these distinct mechanisms of inheritance, and maybe give us new candidates for transcription factors that actually bookmark chromatin. So it's a long answer, but to saying I don't know, but I think it's really an excellent question. Tuba asked me, thank you for your talk. I was wondering how much of these loop extrusions are optimized for recombination during meiosis. Yes. Is this a random process to maximize randomness in recombination between sister chromatids? Yeah, so that's really a really interesting point you raise. And the connection between loop array formation and recombination during meiosis is one actually that started me on this whole thing many years ago when I was in Nancy Kleckner's lab, where she and her students have spent a lot of time studying that. Loop extrusion in meiosis is a little bit more difficult to study, given that we will have 4 chromatids interacting with each other. And the combination of loop extruders that actually make loops in meiosis is still under debate. I think cohesin could be a dominant one rather than condensin. So cohesin could certainly play a role in bringing recombination machineries to the axis of the chromosome, even when breaks are made somewhere in the loop. So there's this interesting feature in meiosis that breaks may happen in the loops, but somehow need to end up at the base of the loop. And again, loop extrusion could play a role in this. We don't know. And also the patterning of events along chromatids is not exactly random. At the single cell level, there is interference between these complexes. So again, I think now that we know the machines that make these loops, these questions can be addressed. The meiosis question is really good, but very complicated for the fact that we have to deal with both sisters and homologs. People ask, amazing, what happens to the cohesive cohesin during anaphase, which hold the loops together? Yeah, very good question. I drew out a scenario where the sisters are connected by cohesive cohesins that hold the loops together. And of course, I ignored the centromere, where cohesive cohesin holds the sisters together at high density. What probably happens during anaphase is that those cohesins get cleaved by separase. So the ring gets cleaved open and the sisters can separate. That cohesin is now lost, but that's not a big deal. Most of the cohesin was lost earlier during the eviction process by condensin. But it's probably a proteolytic cleavage of the cohesin ring. Then Paulina asked, thank you for the talk. Regarding the second story, is it possible that the loop extrusion structures are not visible when nuclear transportation is blocked because CTCF cannot be imported? Ha! What an excellent question. Yes, you are entirely correct that if there's no CTCF, we wouldn't see those dots in the Hi-C map either. I have 2 arguments to say that it is not inability of CTCF import. First, we think CTCF doesn't need import because it reassociates with chromatin very quickly. We have some imaging and chip data for that. And also, Kurt Blobel showed that earlier on. CTCF can bind really quickly during telophase before the nuclear envelope seals. So it doesn't need import. Second, there is additional quantitative analysis you can do on high-C data to show that there are cohesin-mediated loops, even when there's no CTCF. You can get an enrichment of signal, of high-C signal at a range of about 100 kilobase, which is cohesin loops. So we don't see that phenomenon happening in high-C. In fact, we don't see any evidence of any cohesin loops that we should see, even in the absence of CTCF. So it's a very good question, but we have some good lines of evidence to suggest that CTCF is there, but cohesin is not.
That was a great talk. I actually had a question about the nuclear volumes when you block the channels. Why is the volume not changing?
I don't know. It's a very good question. I wish I knew, because to me, this means you need something to decompact chromatin. So condensin loops are gone. We know that from high-C. We know we form chromosomes, interphase chromosome structure. They're just very condensed, or condensed is the word. And the question is, why is that? So you need something else to decondense or decompact chromosomes. And I don't know what it is. We know, I thought maybe it's histone phosphorylation, but the histones become dephosphorylated just fine. So the chromatin seemed to be reset to what it was in interphase, meaning no phosphorylation, no condensin loops, yet the chromosomes are really tiny. Now, one other maybe trivial explanation could be is that these nuclei are not transcribing genes. So there is no RNA, very little RNA being made. And we know that in normal nuclei, in G1 nuclei, RNA actually occupies a significant fraction of the nucleus. So possibly that's just because there's no RNA being produced. I don't know. I like to think that we need something special to decondense the DNA, and maybe we can identify it. But it's a good question. I don't know the answer.
And you guys checked the salts? Like you checked like divalent concentrations and
I don't know how we would do that.
there's
These are intact
dyes.
cells that we're looking at.
There's like dyes. Like you can use like
They should probably do that. For magnesium or cations, bivalent cations in general?
I think there's magnesium specific dyes, and there's also like general divalent dyes. So
It's a good suggestion. We should look at it.
one of the postdocs in Vadim's lab, Cody Dunton, or I guess he's a research professor now, has been looking at like calcium and magnesium, and it's like regulating compaction. I know you have a nice paper from 2022 on that, on the connections being stable.
Yeah. Well,
Yeah.
that's in nuclei.
Yeah.
Then we can modify it. But yeah, no, well, we should probably do this. I will look into that. It's a good suggestion. Something is clearly off and not working well in these nuclei. And the amazing thing is, even though the chromatin is highly condensed, it can form these compartments. And if you wait longer, they get stronger and stronger. So chromatin is movable in this highly otherwise condensed structure. There's a question here from Nathan asking about how does topo2 occupancy contribute to interphase reformation? Yeah, that's a good question too. I mean, we looked at topo2 activity requirements during mitotic exit, and there is a requirement for topo2 beyond just the unlinking of the sister chromatids. And that has to do with probably, we think, removing of remaining intrachromosomal entanglements, and that you need to remove them to form good compartments. Interestingly, in the absence of nuclear import, these nuclei are chock full of topo2 alpha. So in normal G1 cells, the topo2 alpha levels go down quite quickly after cells exit mitosis, but that is not happening in these cells. So topo2 alpha is high in these nuclei. It might contribute maybe to some of these structures we see. I don't know. It's a good question. I think the role of topo2 in interphase chromosome folding is only recently becoming a topic of interest to lots of people.
Okay.
Oh, there's one. Oh, somebody. Thank you. Thank you, Nathan. Thank you, everybody. I know I went over time. We started a little bit late. I had some problems with my Zoom, but thank God I was able to connect to all of you. Thank you for your attention. Thank you for the invitation.
Thank you so much for doing this. That was fantastic, and I'm really glad we got to do some Q&A too. Really good stuff.
Thank you.